Artemia is a genus of aquatic crustaceans known as brine shrimp.Artemia. The only genus in the family Artemiidae has changed little externally since the Triassic period. The historical record of the existence of Artemia dates back to 982 from Urmia Lake, Iran, although the first unambiguous record is the report and drawings made by Schlösser in 1757 of animals from Lymington, England. Artemia populations are found worldwide in inland saltwater lakes, but not in oceans. Artemia are able to avoid cohabiting with most types of predators, such as fish, by their ability to live in waters of very high salinity. The ability of the Artemia to produce dormant eggs, known as cysts, has led to extensive use of Artemia in aquaculture. The cysts may be stored for long periods and hatched on demand to provide a convenient form of live feed for larval fish and crustaceans. Nauplii of the brine shrimp Artemia constitute the most widely used food item, and over 2,000 tons of dry Artemia cysts are marketed worldwide annually. In addition, the resilience of Artemia makes them ideal animals for running biological toxicity assays, and it has become a model organism used to test the toxicity of chemicals. Breeds of Artemia are sold as novelty gifts under the marketing name sea monkeys or aqua dragons. Description: the brine shrimp Artemia comprises a group of 7 to 9 species very likely to have diverged from an ancestral form living in the Mediterranean area about 5.5 million years ago. Artemia is a typical primitive arthropod with a segmented body to which is attached broadleaf-like appendages. The body usually consists of 19 segments, the first 11 of which have pairs of appendages. The next 2, which are often fused together, carry the reproductive organs, and the last segments lead to the tail. The total length is usually about 8 to 10 mm for the adult male and 10 to 12 mm for the female, but the width of both sexes, including the legs, is about 4 mm. The body of Artemia is divided into head, thorax, and abdomen. The entire body is covered with a thin, flexible exoskeleton of chitin to which muscles are attached internally and shed periodically. In female Artemia, a molt proceeds every ovulation. For brine shrimp, many functions, including swimming, digestion and reproduction, are not controlled through the brain. Instead, local nervous system ganglia may control some regulation or synchronization of these functions. Autotomy, the voluntary shedding or dropping of parts of the body for defense, is also controlled locally along the nervous system. Artemia have 2 types of eyes. They have 2 widely separated compound eyes mounted on flexible stalks. These compound eyes are the main optical sense organ in adult brine shrimps. The median eye, or the nauplii, is situated anteriorly in the center of the head and is the only functional optical sense organ in the nauplii, which is functional until the adult stage. Ecology and behavior. Brine shrimp can tolerate any levels of salinity from 2, with an optimal range of 2, and occupy the ecological niche that can protect them from predators. Physiologically, optimal levels of salinity are about 32, but due to predators at these salt levels, brine shrimp seldom occur in natural habitats at salinities of less than 62. Locomotion is achieved by the rhythmic beating of the appendages acting in pairs. Respiration occurs on the surface of the legs through fibrous, feather-like plates. Reproduction. Males differ from females by having the second antennae markedly enlarged and modified into clasping organs used in mating. Adult female brine shrimp ovulate approximately every 140 hours. In favorable conditions, the female brine shrimp can produce eggs that almost immediately hatch, while while in extreme conditions, such as low oxygen level or salinity above. Female brine shrimp produce eggs with a chorion coating which has a brown color. These eggs, also known as cysts, are metabolically inactive and can remain in total stasis for 2 years while in dry oxygen-free conditions, even at temperatures below freezing. This characteristic is called cryptobiosis, meaning hidden life. While in cryptobiosis, brine shrimp eggs can survive temperatures of liquid air and a small percentage can survive above boiling temperature for up to 2 hours. Once placed in briny water, the eggs hatch within a few hours. The nauplius larvae are less than 0.4 mm in length when they first hatch. Brine shrimp have a biological life cycle of 1 year. Parthenogenesis. Parthenogenesis is a natural form of reproduction in which growth and development of embryos occur without fertilization. Thelytoky is a particular form of parthenogenesis in which the development of a female individual occurs from an unfertilized egg. Artemixis is a form of thelytoky, but there are different kinds of Artemixis. The kind of Artemixis relevant here is one in which 2 haploid products from the same meiosis combine to form a diploid zygote. Diploid-Artemia parthenogenetica reproduce by automictic parthenogenesis with central fusion and low but non-zero recombination. Central fusion of 2 of the haploid products of meiosis tends to maintain heterozygosity in transmission of the genome from mother to offspring, and to minimize inbreeding depression. Low crossover recombination during meiosis likely restrains the transition from heterozygosity to homozygosity over successive generations. Diet. In the first stage of development, Artemia do not feed but consume their own energy reserves stored in the cyst. Wild brine shrimp eat microscopic planktonic algae. Cultured brine shrimp can also be fed particulate foods including yeast, wheat flour, soybean powder or egg yolk. Aquaculture. Fish farm owners search for a cost-effective, easy-to-use, and available food that is preferred by the fish. From cysts, brine shrimp nauplii can readily be used to feed fish and crustacean larvae just after 1-day incubation. Instar-I and Instar-II nauplii are more widely used in aquaculture, because they are easy for operation, rich in nutrients, and small, which makes them suitable for feeding fish and crustacean larvae live or after drying. Toxicity test. Artemia found favor as a model organism for use in toxicological assays, despite the recognition that it is too robust an organism to be a sensitive indicator species. In pollution research, Artemia, the brine shrimp, has had extensive use as a test organism and in some circumstances is an acceptable alternative to the toxicity testing of mammals in the laboratory. The fact that millions of brine shrimp are so easily reared has been an important help in assessing the effects of a large number of environmental pollutants on the shrimps under well-controlled experimental conditions. Threatened. Artemia monica, the variety commonly known as Mono Lake brine shrimp, can be found in Mono Lake, Mono County, California. In 1987, Dennis D. Murphy from Stanford University petitioned the United States Fish and Wildlife Service to add a monica to the endangered species list under the Endangered Species Act. The diversion of water by the Los Angeles Department of Water and Power resulted in rising salinity and concentration of sodium hydroxide in Mono Lake. Despite the presence of trillions of brine shrimp in the lake, the petition contended that the increase in pH would endanger them. The threat to the lake's water levels was addressed by a revision to California State Water Resources Control Board's policy, and the U.S. Fish and Wildlife Service found on 7 September 1995 that the Mono Lake brine shrimp did not warrant listing. Space experiment. Scientists have taken the eggs of brine shrimp to outer space to test the impact of radiation on life. Brine shrimp cysts were flown on the U.S. biosatellite 2, Apollo 16, and Apollo 17 missions, and on the Russian Bion 3, Bion 5, Photon 10, and Photon 11 flights. Some of the Russian flights carried European Space Agency experiments. On Apollo 16 and Apollo 17, the cysts traveled to the Moon and back. The cosmic ray passed through an egg would be detected on the photographic film in their container. Some eggs were kept on Earth as experimental controls to ensure a fair test. Also, as the takeoff in a spacecraft involves a lot of shaking and acceleration, one control group of egg cysts was accelerated to 7 times the force of gravity and vibrated mechanically from side to side for several minutes, so that they could experience the same violence of a rocket takeoff. There were 400 eggs in each experimental group. All the egg cysts from the experiment were then placed in salt water to hatch under optimum conditions. As a result, a high sensitivity to cosmic radiation was observed on Artemia Selena eggs. 90% of the embryos, which were induced to develop from hit eggs, died at different developmental stages.